Hi everyone. This is my project. It's about computational analysis of nucleolin microRNA finding homologs. And my mentor was Dr. Singh, who's professor of computational biology at Brooklyn College. And my name is Soha Faruqi. And yeah, this is the project. So the context of my research, so my work revolves around observing nucleolin, otherwise known as NCL, which is a human encoded multifunctional protein. And I use computational analysis to identify NCL homologs and functionally significant residues in conserved regions. So what is the importance of my research? My research is important because identifying the key residues of functional importance will ultimately be helpful in targeting areas that may be chosen as possible targets for curative treatment. This study will also be compared to a past study on the same topic to help discern patterns of evolution and change in the homologs. Okay, and here's some background information. Okay, nucleolin is responsible for maintaining homeostasis within the cell through regulating the various cell functions such as proliferation, survival, and apoptosis. NCL also plays a role in the processing of several microRNAs, or otherwise known as miRNA, which is associated with several cancers, including breast cancer. And lastly, an excessive amount of nucleolin can lead to a reduction in heterochromatin markings on our DNA. Okay. So the overall purpose of my project was to identify conserved residues within the homologs and point out any changes that may have occurred as the organism evolved. And the significance of this research is that it reveals reveals any conserved residues and determine areas that could be used for therapeutic intervention. My main point of research. Okay, sorry. So my main points of research, firstly, all of the organisms were run through NCBI via the BLAST program to find similar sequences in the individual organisms. And from this, the sequences were generated and displayed as their primary structures, along with statistics such as percent positives, percentage identity, e-value, and coverage. And then after documenting these sequences, each organism needed to be processed through several programs to detect similar residue locations for the different RNA binding domains, otherwise known as RRMs, as well as any unique domains that may appear. And you'll see an example of this later on. And moreover, this analysis yielded the information required to start modeling the cartoons for each organism. The cartoons were created in IBS, and they demonstrate the location of different RRM domains on the strip of protein. And lastly, each region of the RRM found in any of the organisms had their individual amino acid sequence listed, and this was compiled onto a list by the Clustal Omega program, which aligned the sequences and combined them in a way that was just more convenient. Afterwards, this information was transported to another program, SPERT, using the BEG feature, so that alignments could be displayed. Yeah. So here are my results of the study. As you can see in this table, these are the homologs that were found in the alignment charts, and here are the organisms that I surveyed and the statistics that I was talking about them. Okay, and this is the domain architecture of the different homologs. They also model the different RM domains on each organism. And as you can see, like I was mentioning before, some of the organisms have unique domains. Like in the first figure, Gallus, you can see that there's a coiled right here. So yeah, some of them have different, and there's another one in figure 10. And then next we'll look at some alignment charts, sequence alignment charts. So we'll start off with the previous one that shows conserved residues from 2021 in a different study. And you can see the conserved homologs are highlighted in yellow on the top if you look here. And down here we have some different residues that are highlighted in different colors, and they show different interactions. Like you can see salt bridges, H bonding. Yeah, you get the gist. And If you, yeah. And then if we go to the next slide, the next few slides are like the newly generated charts that I identified the homologs that were shown like that like from this chart. And it kind of shows which, using these charts, I was able to determine which residues were conserved from the previous study. Yeah, you can see all of them. There's 4 of them. Okay, and then my conclusion. So based on results, it's possible that over time organisms evolve to have specific residues that recognize microRNA molecules, and higher organisms showed a higher level of conserved residues as compared to plants, yeast, and lower organisms that contain NCL. And then here are my references and my thank you slide. Thank you to my mentor, Dr. Singh, and the Talaria Summer Institute for, yeah, I'd like to thank everyone for their support and opportunity.